50% in more or less healthy plants, epigenetic variations are inherited for something like 8 generations. So if this is the case, then we can have, because we have a lot of patterns of methylation within a given genome, there can be many combinations of different variations, and therefore we can have quite interesting and quite important evolutionary change only on the epigenetic axis. Now, we know that we can have cultural evolution, not only in us humans, but also in chimpanzees. And this is just one example out of many. These are two chimpanzees belonging to different populations, and they have different traditions. For example, this is a nut-cracking population in West Africa. There are populations in other parts where they have the same nuts, but they don't have the behavior. And it's not because they don't have a mutation for the behavior. They just didn't invent the behavior for some reason, and therefore it is not transmitted from one generation to the next. And here we have a population which is doing termiting, that is inserting a stick into a termite nest and then taking it out full of lovely, tasty termites and eating them. And again, this is a kind of behavior which is culturally transmitted through social learning, non-symbolic. And differences in behavior which are transmitted from one generation to the next can also lead to speciation. There is a very interesting story about indigo birds who are parasites of some species of finches. And what happens there is that a particular parasite parasitizes a particular bird. That means that the parasitic bird lays its eggs in a nest of another bird, another species of bird, and this host bird is looking after the little ones. It doesn't know that they are not its own, and the basis of this kind of parasitism is very interesting and can be manipulated. What happens here is that once the indigo bird lays its egg in a host nest, the little ones, the little chicks, learn the song of the host species. And the females prefer the song of the host species in which they were raised, and therefore they will choose males which sing this song. So the males learn to sing the song of the host, and the females prefer this song. Now, if we can take an egg and switch it into a nest, into a host species which it never saw, suddenly we will have a parasitic bird where the males prefer, when the males sing a new song and the females prefer this song. So, and they will not mate with, the females will not mate with males who sing an inappropriate song. So in one generation, we can have reproductive isolation. We can have a situation whereby, because of behavioral criteria, a particular bird will only mate with those who sing in a particular way. And in this way, a new population can be formed, which is reproductively isolated from the original population. Now, one of the very interesting things that has been found was that that it is not only environmental changes, but also genomic stresses that can lead to very big, very important changes in both DNA and in epigenetic factors such as DNA methylation. So what we have here is a population of plants, and there can be all kinds of stresses. For example, an ecological upheaval, a very strong bottleneck, which is causing the population to start from very, very few or even one individual. Polyploidy, which is a duplication of chromosomes, or hybridization. When two species hybridize, they form a hybrid, and this can be very often, this can be accompanied, if the number of chromosomes is unequal, by duplication of chromosomes, allopolyploidy. Now, all these kind of things, these very, very strong stresses, lead to an upheaval, to a kind of revolution in the plant genome. Transposable elements, all kinds of genetic elements, start jumping around. The pattern of methylation can be very, very drastically changed. 13% of the genome in wheat, after following hybridization, changes its methylation pattern, and these things can be inherited. So what we have here is a very, very big change, a great variability, genetic and epigenetic, which is brought about by great stress, and this can be then selected. So the epigenetic mechanisms are not only mechanisms that are important for microevolution, they can be also important during those periods where there is a very big macrovariation, creation of macrovariation. So we can think about microevolutionary changes and macroevolution. Now, medicine is very important for us, and we would like to see how all these things that we have learned are important for how we can treat disease, how we can get healthier. Now, what we know is that epigenetic inheritance, cellular epigenetic inheritance, that I was talking about in the first part or third of the lecture, is important for understanding many types of cancer, some complex diseases and environmental diseases and age-related diseases. Now here we are, what we have learned about the relationship between inherited patterns of methylation within the body and cancer. Now we can have tumor suppressor genes, genes that inhibit the formation of cancer, and it is important that these genes will be active, and they normally are active. But these genes can become, for whatever reason, methylated, and once they are methylated, this is transmitted. There is no inhibition of cancer formation, and therefore the chances of cancer developing are much, much higher. And here we have the opposite. We have oncogenes, which are genes whose activity causes cancer in the wrong place at the wrong time. And usually these genes are heavily methylated, so they are not expressed. But if they are demethylated for whatever reason, and that can be as a result of nutrition, of stress, of all kinds of things, of age, this situation is inherited, and the chances of cancer are greatly enhanced. This is very important for several types of colon cancer, for example, but not only. Now, aging. We know that aging is a very, very complex phenomenon, but we know that one of the things that happen during aging is a change in DNA methylation of many, many genes. And we also know that food can lead to changes in DNA methylation. We know that people who are living in very poor conditions there are all kinds of, the offspring that they bring into the world are usually smaller and thinner, and they have a predisposition for many diseases in later ages. And we know from work on rats where this kind of system has been simulated, that there are changes in DNA methylation in very important genes which are related to the functions of the liver, to cardiac functions, and so on. And here we have a nice example from rats. Now, these two rats, which look these two mice that look rather different, have the same DNA sequence exactly. And the reason that they are different is dependent on the food of their mother. This fat yellow mouse is bigger, fatter, and tends to have tumors and diabetes and all kinds of problems. And this is a normal little looking mouse, healthy, happy, so on. But they're genetically identical, exactly the same. And here we are. They are what she eats. Now it's a particular strain of mice that we're looking at. In this mouse had a normal diet, and her offspring, most of them were yellow, and she had some brown mouse. And here is the same mother from the same pure line with exactly the same DNA, or even the same mother, but this time given folic acid. And what happens now is that the number of normal brown mice that she brings into the world is proportionally higher. So, and we know in this case also what the gene is And we know that it is a difference in methylation that makes the difference in the activity of the gene. Now, okay. So what are we to conclude? There are many, many other aspects that, of course, we can talk about, but I think that what happens is that because we are thinking about heredity in a wider way, and we're thinking about mechanisms of heredity, of transmission, that are at the same time also part of regulation, we have to expand our notions of heredity and evolution. They have to be not only defined like, as Dobzhansky suggested, that evolution is a change in gene frequency. This is not sufficient. It's okay. There's nothing wrong with this, but it's too narrow a definition. So we have to think about heredity and about evolution in a wider sense. And it has many, many broad implications, some of which I touched here, that are important from a theoretical point of view and from practical points of view. Thank you very much. Thank you.
Thank you very much for this interesting lecture. Any questions at all?
I hope you have.
Yeah?
I would like to ask you about your opinion concerning this hypothesis of the expansion of evolutionary and hereditary theory in biology. It is interesting, I noted that this extension brings us back to some pre-Darwinian concepts of evolution, like Lamarckian view, the heredity of acquired characteristics. And my first question is whether you think that the inclusion of epigenetic levels and, I don't know, genomic regulation, metabolic regulation opens some new space for the returning to another pre-Darwinian concept of structuralism in biology. You know what I mean, about the laws governing organismic structures, their dynamics, and morphologies.
Okay, so you have two questions really about Lamarckism and about the structural approach to biology, which is not exactly the same. Yes. Okay. Yes, I think the answer is yes. I think that in some ways we are Lamarckism was a very dirty word in biology for a long time, in evolutionary biology. And it was partly because Lysenko made such a terrible abuse of genetics and was a tyrant and charlatan. And, but yes, there are aspects. I mean, you know, the inheritance of acquired characters in a way is an unfortunate phrase. It's the inheritance of developmental predispositions maybe that is happening. And these epigenetic mechanisms show how it is possible. We see in the case of this mice that got the great-grandmother got the vinclozolin, their father, the first generation was affected by this. It affected the embryo in utero. And then this thing that happened to their great-grandfathers was transmitted through the generation. Not the trait, but the developmental predisposition for the trait, the methylation pattern on particular genes. So yes, I think that we are